It's my pleasure to introduce Dr. Aaron Lau, who will be speaking on sequence-specific peptoids for self-assembly and biomedical applications.
So thank you, Alex and Ian, for inviting me. Yeah, thanks again. It's really nice to speak to a diverse and multidisciplinary audience. You will be hearing a lot of chemistry in my talk. This is a switch in gears, very much so actually from the last talk. We are going to deal with chemical building blocks and design materials from the ground up. Just a word of introduction about the University of Strathclyde. We are in Glasgow, and actually we occupy a significant chunk of the downtown city area. We have about 200-year history. It's a good-sized public university in Scotland, 15,000 undergraduates. Chemistry department has 650 undergrads and 200 PhDs and postdocs. Our first chair, Thomas Graham, discovered one of the laws of diffusion. So the metrics say we're gaining in prominence. That's very nice. But what's perhaps nicer is that the university has an estate next to Loch Lomond, and the department sometimes rents it out for internal events. Anyway, my research is in bioinspired molecular nanotechnology. What I mean by bioinspired molecular nanotechnology is that when we take molecular principles in biology and try to create materials that mimic or surpasses the functionalities observed. So that's the bioinspired part. And the molecular nanotechnology part is, well, what do we look at in nature, right? If you look at a cell, for example, what you will see is a hierarchy of structures from the, for example, collagen fibers that tie cells together to the proteins, the nanoscopic proteins on cell membranes that essentially control cell interactions. Underlying all of this hierarchy are actually sequence-specific polymers. So polymers, as you know, are long chains of molecules, or we call monomers, joined together. And sequence-specific polymers are those that has a whole bunch of different relatively simple monomers arranged in specific order, one after the other. And this covers all DNA, the ACTG genetic code. So those are the 4 monomers of DNA, and they are in specific sequence that gives you genetic code. And also of peptides, you have about 20-odd amino acids. And it's that sequence that generates the chemical and physical functionalities that forms proteins. If you didn't know, I'm telling you, proteins are the building blocks of life. And essentially that's a kind of materials grand challenge. How can we make or engineer materials that are as diverse and functional as proteins, right? So making proteins is really hard, and lab doesn't try to make proteins as such. In fact, we try to do something hopefully simpler and more controllable. So we work on what's called peptoids. So these are mimics of peptides, mimics on the molecular scale. So like peptides that are made up of amino acids, as I was saying. And so this is a chemical structure of a peptide. Just to jog your memory, if you had ever taken chemistry class, or if you haven't, okay, this is it. This is a molecule, and it's indicated in these chemical structures by lines that represent chemical bonds between atoms. So carbon atoms, hydrogen atoms, oxygen atoms, of nitrogen atoms, CNO. Those are the fundamental elements of organic molecules. And amino acids are differentiated from each other by what we call the side chain R group. R group just means a arrangement of atoms. And these are some example R groups. And each amino acid has a different R group. And so when you arrange them in specific order, these amino acids with different side chains, then you get your peptides and your functional molecules. Peptoids are completely synthetic versions of peptides. And what we do here is actually we shift the attachment position of this R group. And what this does is actually it eliminates a very special hydrogen on the nitrogen here that forms a peptide bond. This hydrogen is actually what makes proteins form. It contributes to something called hydrogen bonding. Taking it out simplifies the intermolecular interactions by quite a lot. And instead, so we concentrate all the functionalities on the side chains. So that is essentially the beauty of peptoids. Second beauty of peptoids is that you can, well, with the magic of modern chemistry, you can install essentially any synthetic side chain you want. So while in nature, we build it truly from the ground up multifunctional peptides from simple functionalities of these natural side chains, we can directly put in function by choosing a synthetic small molecule and just directly put in a function. So that's pretty neat. But we can also do what peptides do. We can use very simple side chains and build up function from the sequence ordering of these building blocks, monomers. Another beauty of peptoids is that, well, because now they're synthetic, so they are not recognized by enzymes. Remember, we eat proteins. Proteins are made of peptides. Peptides essentially then get degraded by natural enzymes in the body. So if you want to have a drug that's a peptide, you can't eat it as a pill. It would just get degraded. Peptoids have this advantage that they're not degraded. And so they are, in that way, more stable and more active as a biomolecule. So we'll go through some examples of how we explore this ground up building of materials from peptoids. We'll look at some examples of self-assembly and then some biomedical applications. Peptoids are still relatively expensive, so we go for high value type of applications. So you can even if you can just produce small amounts of peptoids, it'd still be useful. And self-assembly, of course, is part and parcel of how biomolecules work. And we do want to learn the rules of self-assembly so that we can produce structures and other functionalities. And these topics also illustrate the structure of my lab. So we are centered around building this bioinspired polymers, but we also do a lot of work in attaching different functional groups together, so bioconjugation or functionalization. And we also have a lot of activity in characterization. When you build a nanomaterial, you need to make sure that it's what you think it is. So first topic, self-assembly. You hear about it a lot, I think, this term self-assembly. So on the molecular scale, molecular self-assembly, what is it, right? And there are two groups of inspiration for self-assembly. One is what you see almost every day, bubbles, surfactants, soap. You rub soap together, you form bubbles. That's, in fact, on a molecular scale, self-assembly, and is controlled by hydrophobicity. So basically the concept of how oil and water don't mix. So they want to stay apart. Molecules that are oily or hydrophobic and molecules that are water soluble or hydrophilic. These two parts want to stay apart, but in a surfactant, what you do is you join the hydrophobic, let's say this tail group here, together with a hydrophilic group, a head group. So they have to be together, chemically bonded together, but they do want to stay apart as well. So these two parts of the molecule want to stay apart from each other. And the oily parts also want to stay away from water. And so what happens is that if you have a whole bunch of these, what we call hydrophobic, hydrophilic, or what we call amphiphilic molecules together, then all the oily bits or the hydrophobic bits tend to aggregate together. And then the hydrophilic bits help solubilize these droplets. These are actually emulsions. So this is known for a long time. And if you vary the shape or the relative length and shape of these, then you get different shapes, just so that these molecules can pack together. So that's a simple part of what we can understand. The other part of what nature does is it pushes these principles to the extreme and specifies sequences that are that have very high definition or fine-grained control of where the hydrophobic and hydrophilic bits are, and and you have a lot of these bits of these sequence-specific polymers, peptoids or peptides, joined together, and you can even have protein folding. So from a linear sequence, you have a 3D shape. I think that's quite marvelous. Also observed just shorter sequences that we have found in nature, like in amyloid aggregates or fibers that contribute to neurodegenerative diseases. We know of sequences that drive this self-assembly. And this, for example, KLVFF80 sequence, denoted by the single letter code of amino acids, they're known to form fibers, and people can mimic these sequences, just synthesize it in the lab and combine principles of a surfactant together and form these fibrils. What we want to do with peptoids, now that we have that fundamental understanding, is to, well, try to recreate that in a way. Knowing something is one thing, in principle. Being able to apply it, if you can do that, actually demonstrates another level of understanding, right? And in the process of trying, you gain more insight. And so one of the activities in the lab is just to see how peptides self-assemble, especially because now we say we have simplified the interactions. So we've taken out some of these interactions that could help with self-assembly. And we say, can we just use the side chain chemistry to enable these self-assembly? Now, just a side note, again, a word about the chemical structures. Now, for better clarity of these diagrams, I've taken out the symbol C for carbon, and each vertex in these structures is a carbon atom. So just for clarity, this is a simplified view of the chemical structures. And what we have are, well, in our recent paper, we call minimal self-assembling peptoids. What we're saying is that we have taken this diphenylalanine motif and say, okay, let's see what happens in peptoids which do not have these hydrogens there, this hydrogen bonding in the backbone, in the main chain. Can they still self-assemble, basically? So this is a set of 6 peptoids synthesized in the lab. Let's see what happens. So the first thing is, okay, the simplest one is just 2 peptoids, 2 monomers, the equivalent of FF. First roadblock is that they're not soluble. This is hydrophobic groups, and they do crystallize into beautiful shapes. So as a fundamental study, it was beautiful. But for applications wise, it's not very useful. Anyway, we were able to obtain the crystal structure of these solidified dipeptoids. And so what we are seeing is that now to crystallize, they're still hydrogen bonding in the ends of the peptides. So the molecules essentially went that way, joining end to end. In order to make it a little bit more biologically relevant, we added the third monomer, which has this NH2 amine hydrophilic group. This actually becomes charged. So for the first time now for peptoids, we also can generate these fibers that are kind of reminiscent of what we showed before with the amyloidal fibers. But this is very sensitively controlled by the exact side chain that we use. For example, if you extend this side chain by just 2 carbon on the side chain, so 1, 2, 3, 4 carbons on this side chain and just 1, 2 here, the shape, the packing of these molecules change completely, and you don't get these uniform fibrils anymore that can bundle together. Instead, you get these blobs, not very well defined. And then if you reposition where we attach this amine side chain to the end now, then again, you just get different structures. So the position, the sequence of these monomers are really important, just like what we expect from biology. Okay, we can demonstrate that even without the extra hydrogens on peptoids, we get self-assembly, and we can use similar assembling motifs as in peptides to guide the design of peptide self-assembly. Can we also incorporate some of the ideas of surfactants for peptide self-assembly? And so this is what we set out to do with my collaborator, Ian Hamley at Reading. We said, okay, so if we had a head group of peptoids, what if we added a hydrophobic chain at the end of it, right? So this hydrophobic chain should want to gather together, and that might drive the self-assembly. In a way, it's a no-brainer, but nobody demonstrated it before we published this, so let's try. Indeed, we were able to see, this is in transmission electron microscopy, we were able to see these small, around 6 nanometer wide objects, and in at least 3 of these designs. And then when you change the sequence again, you get a slightly different morphology, and you control the size as well. And to know whether these are actually self-assemblies, whether we actually have the hydrophobic core, we used what we call a fluorescence assay, a pyrene test. Basically, we use a fluorescent hydrophobic molecule, pyrene. And if these things are around, then this hydrophobic molecule, which doesn't want to stay in water, will also sequester inside the core of these self-assemblies. And indeed, we can, by tracking the amount of peptoids in the solution, we see that once the amount of peptide rises above a certain concentration, these might sort of start forming, and then pyrene, which also fluoresces brighter within these hydrophobic cores. You can see this increase in fluorescence. Then we see, oh, okay, so above these concentrations of peptoids, then you have these assemblies. Below which, no, they're just single molecules. They haven't come together yet. We also do small angle X-ray scattering and dynamic light scattering to try to measure more accurately the size, not just looking at a few examples in an electron microscopy image, but a whole solution full of these molecules. And these scattering intensity, you can curve fit them, and you can work out what the sizes may be. So basically, this curve would be if it were 80 nanometer sized aggregates, and this curve here in solid black would be the 2.8 nanometer size assemblies. So these are P1 and P2, 2 different peptide sequences with the same tail, and you can see that there is definitely, we can control the size of these things as well. So one of the reasons why we wanted to do the hydrophobic tail self-assembly with peptoids, and we call those lipopeptides, as in lipophilic or lipid plus a peptoid. Why we wanted to do these lipopeptides, it's the idea of making the self-assembly more universal. As in, what we really want is to take any bioactive peptoid, and by doing something simple, like adding this hydrophobic tail to it, doing something simple and make it self-assemble without having to think too much about it. There are so many possibilities, it will be impractical to go through all of them. So based on the previous study, I say, okay, so lipopeptides work. Let's also try to add a tail to these antimicrobial sequences. So these sequences on their own can kill bacteria. It's great, except that they also kill mammalian cells. That's not so great. So then this sequence, for example, is very potent, and it's discovered about 20 years ago, not on the market, not in the pharmacy, because yes, it also kills our own cells, not just bacteria. And the idea is that if we can somehow package these active molecules together, can we actually control both the toxicity and the bactericidal activity, right? And generally, we want to limit environmental release. We want to limit systemic toxicity. Coming up with a toxic or bioactive molecule is actually quite easy. Coming up with one that's bioactive and doesn't kill you, and also doesn't contribute to environmental release, it is the hard part. So micellization or self-assembly packaging is one way that we went about it. And attaching to surfaces, which I won't talk about today much, is another way to go about it. So that's what we did. We again came up with a set of peptoids, and now we're changing the tail length. We are saying, okay, are we going to add or don't add a cationic group or not? And let's see, yeah, do they self-assemble still? Are they still bactericidal? Are they still toxic to our body? So this is some data of what happens when you add different species of peptoids to solutions of different bacteria. These are bacteria that you would see in a hospital setting pretty often. And we vary the length of the peptide itself, this length, as well as the lengths of the tails. Right? So high numbers is bad. High numbers means that we need a lot of this to kill the bacteria. We don't want that. We want small numbers. So small numbers is good. What's even better is if we can, from a original without a tail, large number, if we can decrease it, we can make it more potent against the bacteria. That's good. That's what we want to see. Right. So indeed, we show that we can do that by adding a certain tail length. And this depends, however, on whether the length of the peptide itself is on a certain length. We also show now that indeed we can self-assemble. So critical aggregation concentration is that concentration I showed before, at above which it starts to self-assemble. So again, lower CAC is what you're looking for. It's good because it means that you need fewer of the peptides in order to self-assemble. So straight lines are always good in an experiment too. So we see that by adding certain components, we can actually achieve the self-assembly. And in fact, we also change the size of these assemblies as well. So we have self-assembly, we have increased, we can enhance antibacterial activity. What about toxicity, right? So the longest peptide has this very low viability of mammalian cells. So low number in this case is bad. So we want high viability of mammalian cells. But you can see that without the tail, although it's also toxic to bacteria, but that's of no use. But what we're showing here is that by C8, we can actually enhance the viability by quite a lot. So that's good. So, and for these shorter peptoids, yes, this has high viability originally, but it's also of moderate activity against bacteria. But by adding certain lengths of tails, we can maintain the activity and increase the viability as well. So the conclusion is that, yes, we can use these components to control both toxicity and activity and self-assembly. I don't have enough time today to go through all the examples in my lab, but we also do somewhat more complicated peptoids. We can form sheets of them. And again, we use the hydrophobic fluorescent dye to indicate whether they have assembled or not. Okay. So we feed them to stem cells, and they're not toxic at certain concentrations. And in fact, some early studies that we are doing now show that they are controlling differentiation. Here, we also attach peptoids on surfaces. It's actually a very useful thing to do. We can repel bacteria and other organisms. So again, because we have sequence control with peptoids, we can control the spacing and the charge distribution. And we study how proteins interact with the surfaces. By controlling the spacing between the peptoids, we control the amount of proteins attaching to the surface. Other than just the sequence, we also are mindful that other things around the peptoids can also be important. So when we attach this antimicrobial sequence to the surface, we found that, oh, we actually need a spacer. We need a long chain of another polymer in order to change the characteristics. So this is red means dead bacteria, basically. Green means happy live bacteria. So just glass, this bacteria attaches on it. If we attach the peptide directly, we have some dead bacteria indicated in red and some still happy ones in green. But if we space it apart from the surface a little bit, we get we get more killing. So to conclude, in my lab, we're using sequence-specific peptoids to explore the fundamentals of self-assembly and also to design sequences that can have bioactivity. And the usual experimental procedure is we observe the sequences available in nature. And we say, okay, these features, this type of ordering of monomers probably will give you this type of activity, or we know of physical rules or chemistry rules to say, okay, if you have these chemical functionalities, then it should have such properties. Let's combine it. Does it self-assemble the way we want? Does it have the activity that we want? And it's screening through these small sets then teaches us about how to construct properties that we want. So with that, thank you for attention. I thank my group and the funders, and I'll be happy to take your questions.
Thank you, Dr.